The influenza virus is an important RNA virus. It is a negatively stranded RNA virus in the orthomyxovirus family. It is the only important orthomyxovirus for step 1. The virus has two important surface proteins, hemagglutinin and neuraminidase. These proteins are responsible for entry and exit of the virus into cells. Hemagglutinin is the surface protein responsible for binding sialic acid sugars on respiratory epithelial cells, facilitating entry of the virus. Hemagglutinin makes the virus say hello. Neuraminidase is the cell surface protein responsible for cleaving the sialic acid residues, facilitating exit of the virus from the cell. It says, "Nice to meet ya," as the new influenza viral progeny leaves the cell. Do you remember the names of the neuraminidase inhibitors that can be used to treat influenza? That is oseltamivir and zanamivir. Very good. One of the reasons that flu infections are so dangerous and why we spend so much time trying to vaccinate our population is because it can lead to the development of what is called a bacterial superinfection. And do you remember the 3 important bacteria that can cause a pneumonia during influenza infection? Staph aureus, Strep pneumoniae, and H. influenza. Very good. So why is it that influenza predisposes our patients to bacterial pneumonia? Well, let's go to the whiteboard and talk about why. So here we have part of the respiratory tract with the respiratory epithelium. Just imagine these cells right here. These cells have very important processes called cilia. Cilia are finger-like projections that exist on the cell that are responsible for sweeping the bacteria up and away from the lungs and protecting our lungs from infection. When influenza infects these respiratory epithelial cells, we get swelling and edema of these respiratory cells that leads to impaired ciliary function. You can imagine now these cilia are less effective because of this edema, and therefore the bacteria have a better ability of descending down into the lung. So without good ciliary function, patients with influenza are more susceptible to bacterial infection in the lung. The influenza virus has some interesting and high-yield genetics associated with it. The influenza virus is unique and dangerous because it has the ability to mutate regularly. There are 2 mechanisms by which the virus can change its genetic makeup: antigenic drift and antigenic shift. Antigenic drift describes the process of mutations in the hemagglutinin and neuraminidase genes, leading to minor changes in the virus's protein makeup. This sort of genetic alteration is dangerous because our immune system doesn't recognize the virus even if we've had the flu before. These genetic changes are why we have a new flu season every year and can lead to epidemics or intense regional spread of the virus. However, even more dangerously, the influenza virus is capable of undergoing something called antigenic shift. Remember, the influenza virus has 8 RNA segments, and therefore it is able to undergo genetic reassortment of those viral segments. Genetic reassortment occurs when influenza virus A and influenza virus B infect the same cell. In this one new cell, there are now 16 gene segments. Remember, each virus has 8 segments. Segments of virus A can exchange with segments of virus B and create a new virus C, seen here. This unique progeny of virus A and B has genetic material from both virus A and B and is going to be much different from its parents. Because this is a big change to the virus is now extremely unique and unrecognized by pre-existing immunity. This can result in worldwide pandemics. So in summary, antigenic shift is more serious than antigenic drift because it results in exchange of entire genetic segments rather than simply mutations in the genetic material. A way I like to remember this is by thinking of how some cars drive on the freeway. As I'm sure we all have seen, some cars drift in their lane, maybe swerving a little bit from one side of the lane to the other. These are minor changes or drifts in car position and can present some danger to us as the driver in the adjacent lane, for instance. But then there are also drivers who shift lanes really fast, making serious changes or big changes in their car position and nearly smashing into us as they do it. This dramatic shifting of lanes is a bigger change and poses a bigger threat to us driving in the next lane than the guy who just was drifting a little bit in the lane next to us. I hope that can help you remember the differences in how serious antigenic shift is versus antigenic drift. So let's do another flash quiz. Which influenza virus surface protein is responsible for facilitating infection of target cells? Facilitating infection of target cells. Remember, it's hemagglutinin, which says hello, and neuraminidase facilitates exit of progeny from the cell, and it says nice to meet you. Remember, these are the two important surface glycoproteins found on the membrane of the influenza virus. These are the proteins our flu vaccine are protective against. Also, we have neuraminidase inhibitors that are effective against the flu early on in the infection. Those are called oseltamivir and zanamivir. Very good. Rubella virus is a toga virus, or a positive-stranded RNA virus that causes a disease called German measles. Patients with German measles have symptoms such as fever, post-auricular lymphadenopathy, arthralgias, and a fine maculopapular truncal rash that begins on the head and neck and spreads down, as we see in this child here. In children, German measles is a mild disease that self-resolves. Rubella is a really dangerous infection if it is acquired by a woman during their pregnancy. It can readily cross the placenta and cause a severe congenital disease. In fact, it is actually the R in Torch's infections. And do you remember the characteristics of congenital rubella syndrome? It can lead to deafness, blindness, heart and brain defects, as well as a diffuse petechiae, as we see here, also known as that blueberry muffin appearance. Luckily, we have a vaccine for rubella. It is part of the MMR, or the measles, mumps, rubella vaccine. The rubella portion is a live attenuated virus. So now let's discuss the paramyxovirus family. Do you remember the 4 viruses that make up the paramyxovirus family? That includes the parainfluenza virus, the respiratory syncytial virus, measles virus, and the mumps virus. Very good. Do you remember what kind of viruses they are? They are negative-stranded RNA viruses. Very good. These viruses are most important causes of respiratory infections in infants and children under the age of 5. Before we talk about each of them individually, there are a few important concepts shared by all the paramyxoviruses. All of the viruses contain a surface F or fusion protein. This is a key infectious protein that not only assists in infection, but causes infected respiratory epithelial cells to fuse and form multinucleated cells. These multinucleated cells are called syncytia. And this is why that RSV is called respiratory syncytial virus. If we remember that RSV is a paramyxovirus, we can remember that all paramyxoviruses form syncytia, which are multinucleated fusions of cells that form because of this unique F or fusion protein. For respiratory syncytial virus, which can cause severe bronchiolitis in infants, we can give a monoclonal antibody called pembrolizumab, which is a monoclonal antibody against the fusion protein, which prevents bronchiolitis and pneumonia in premature infants. Croup is a common infection seen in the pediatric population. It is a term used to describe inflammation of the upper airway. The upper airway includes the larynx, trachea, and bronchi, which we can see in the name laryngotracheobronchitis. Inflammation of this area leads to narrowing of the air pipe, causing a seal-like barking cough with an inspiratory stridor. This tracheal narrowing also leads to a characteristic sign which is seen on X-ray called the steeple sign. If you look closely at the X-ray image, we can see a steeple formed here that's caused by the edema and narrowing of the upper airway. The inflammation is caused by the parainfluenza virus. Severe enough disease can result in complete obstruction of the upper airway, resulting in pulsus paradoxus. The measles virus, also called rubeola virus, causes measles. Measles is an important virus to know for step 1 because it has a lot of very classic and testable information, including the viral prodrome that is commonly referred to as the 3 C's, which include cough, conjunctivitis, and coryza. Coryza is a term to describe a head cold. Personally, I like to add a 4th C, which stands for Koplik spots. I know that Koplik spots starts with a K, but it's easy to remember since C and K sound the same here. Koplik spots are 1 to 2 millimeter blue-gray macules that appear on the buccal, gingival, and labial mucosa within 2 to 3 days of the onset of symptoms. This image is very high yield for step 1. If they show an open mouth of a sick child, immediately be thinking about the measles virus. After the development of Koplik spots, the characteristic rash of measles begins to appear. This rash, as seen here, is an erythematous maculopapular rash that typically begins at the hairline and spreads downward. Think about pouring red paint over somebody's head and watching it spread on the head and face first, and then spread down onto the shoulders, trunk, and all the way down. Complete recovery from measles generally occurs within 7 to 10 days from onset of rash, compared to a 3-day rash that we see in rubella. Vitamin A supplementation has been found to reduce mortality in measles, but the best way to not get sick from measles is to prevent it altogether with the MMR vaccine. This vaccine is a live vaccine that protects against measles, mumps, and rubella. Uncommon but severe sequelae of measles can include post-infectious encephalomyelitis and subacute sclerosing panencephalitis, also known as SSPE. SSPE is a chronic progressive encephalitis that causes inflammatory white and gray matter lesions of the brain that occur 6 to 15 years after apparent resolution of measles symptoms. Symptoms of SSPE include neuropsychiatric changes, seizures, coma, ataxia, and myoclonus. Mumps is another paramyxovirus which is acquired through respiratory droplets. Mumps infection is characterized by inflammation of the testes, also known as orchitis, causes inflammation of the meninges, also known as meningitis, and finally, it causes inflammation of the parotid glands, also known as parotitis, as we see here. If we look at the letters that start each of these inflammatory conditions, we form the word POM. We can remember that these inflammatory states occur as a result of mumps virus infection by remembering that mumps virus makes our parotid glands and testes as big as pom-poms. The mumps virus is the last virus protected by the MMR vaccine. Now time for a flash quiz. Which monoclonal antibody can be given to children with RSV infection? That would be palivizumab, the monoclonal antibody that binds to the F protein on the RSV virus, helping our immune system fight off the disease.